Yes, sir.
All right. Thank you. Thank you. All right. So we will go into the class today. Today, what I thought, I will introduce to the concept of PCR. Right. PCR is something that we have taken it for so granted, and it has played such a vital part in the current epidemic, right? Current pandemic, I would rather say that PCR diagnostics has been like one of the biggest, boon or the biggest scientific this thing feat, right? Routine, RT-PCR has become so routine that people don't really pay too much attention to it. But we will, all of you are aware of how PCR works. So I will have a very short, there are several slides, but I may not be spending time on all the slides because some of them are like overlapping. Okay. And then we will get right into the real-time PCR, how it works. And there are several advanced versions of these PCRs, which go right into quantifying, which may be very, very useful under certain circumstances. We'll probably look at that also. And then, maybe either in today's, I doubt if we'll be able to complete everything in today, but definitely this and maybe the next class, we will try to finish the PCR module. Okay. Okay. Let me share my screen. All of you must be aware of PCR, I guess. Or is there anybody who is not aware of PCR? It's okay. I mean, like if you're not aware, I can understand. But all right, all right, all right. Is my screen visible?
Yes, sir. Yes, sir.
Okay, okay. I'm just putting on. Okay, so what we are going to discuss today quickly is basic principles and components of PCR, right? And okay. So the if you look at the trend, if you go to PubMed and say PCR, this of course is way old, but what it shows is an exponential increase in the use of this technique. This technique is so simple, so this thing people are baffled. Why people didn't think of it, although there are some reasons to believe that people try to do this, but it is just that the timing and the technology were not appropriate. Okay, so it didn't happen. What should have happened probably in the 50s, but anyway, so this was PCR became kind of this thing commonly accepted tool in the late 80s, and then in a matter of few years, the technique had become so advanced that people were trying to spend a lot of time in optimizing PCR and making sure that PCR becomes a acceptable, universal acceptable technique. Today you can go to any part of the world and you will see a PCR machine, and in some form or the other PCR is being used for many different activities. Diagnostics is just one thing that I said. The current pandemic PCR diagnostics is something that is so routinely used, right? So just to show that the popularity of this technique is so this thing that there was an exponential explosion of this. Okay, the first scientific publication that came out on PCR was in 1985. It came in a magazine called Science, which is like one of the top three journals in scientific publication. Never use the word PCR. Never use the word PCR, right? So PCR, as such, this term was coined little later. But if you look at it, the the title of the paper said enzymatic amplification of beta globin genomic sequences, and the analysis for the diagnosis. So the first paper itself talked about diagnostics using PCR. Okay, and this was published from a company called Cetus Corporation. So the person who worked there, Carrie B. Millis, he was the one who entirely did the whole work, and he had a couple of other people, and so this got published in this magazine called this thing. Okay, so there was no this thing. I will probably send out article by Carrie Mullis where he discusses about how he discovered this PCR process, how he rationalized the entire process and how he went about doing it, and what are all when you do something in a company, unlike in an academic environment where it is only publication that matters, but when you do it in from a corporation, from a company, the company looks at it from a very different perspective. So he had to get stuff signed by the patent attorney, so many different things. So it's a nice this thing, which is also a prelude to what you all will be reading about intellectual property rights and other these things in your next semester. Okay, so everything that we do in science has we have the ability to monetize or make money out of some basic stuff that we do. Okay, and any intellectual property that comes, it belongs to the person or to the organization. It depends on the situations, right? And you have the ability to monetize or make money out of it, but that's not the part of this thing craze talk, so we're not going to discuss about that. Okay. Okay, so what is polymerase chain reaction? I'm sure all of you have had exposure to polymerase chain reaction from your senior school. So it is a method of in vitro cloning, right? What we are trying to do is to clone a single piece of DNA into X number of copies. We will know what this X number of copies a little later. So it basically allows for amplification of specific DNA molecules in vitro through cycles of enzymatic DNA synthesis. And this is one of the most popular and widely used technique in all the in today in the biological sciences. Why is PCR so popular? Because it is extremely simple, it's extremely powerful, it is sensitive, specific, and reliable. If it were to be sensitive, specific, but if it is not reliable, then any technique may not be widely used, right? Or if it is reliable but it is not specific, then again it's a problem. Okay. And the third most important thing is this extremely fast. Gone. See, when PCR was done, people would spend half a day, trying to do many different things, and then end of the day they will get a PCR result, whether it is positive or negative. But today we know that, in a matter of 5 minutes, we can get a PCR result. So things have changed so much. So that is one reason why PCR is so popular across the globe. Okay, so what is the purpose? The purpose is to clone or duplicate any given molecule, and the principle on which PCR works is it separates the double-stranded template. Okay, the which serves as this thing starting point for DNA synthesis. You include certain short DNA fragments called primers. Many people call it as primer very popularly. Is technically it is an oligonucleotide, but there is not it is a matter of semantics. Some people call it as primer, some people call it as oligo. Okay, initially people used to call it as oligos. Okay, forward oligo, reverse oligo, but nowadays people simply call it as forward primer and reverse primer. So they are the same. Okay, and then once you allow these complementary oligonucleotides or primers to bind to the regions of interest, you basically extended the or synthesize new strands of DNA synthesis by DNA polymerase along with your DNTPs. Okay, the 4 DNTPs. And in certain cases, you may have other proteins also involved along with DNA polymerase under special circumstances. Okay, so that's the principle by which it works. What is the purpose of a PCR? The purpose of a PCR is to amplify double-stranded DNA molecules which will be identical in size and sequence by enzymatic method and cycling conditions. So this is why it is called as in vitro cloning. What do you mean by in vitro cloning is that you have created identical copies of a given gene sequence. Okay. Um, so how does it work? It initially starts with denaturation of a double-stranded DNA template, and then you kind of bring down the temperature so that the primers or oligonucleotides can go and anneal to these specific sequences. Okay, and then you have extension of the double-stranded DNA molecule. So that is how you end up doing the polymerase chain reaction. Okay. Now, typically, when you melt the double-stranded DNA, it all depends. When you take a genomic DNA is highly complex. Suppose you want to identify, a small sequence, small bit of sequence from a genomic DNA. You can imagine, let's say, E. coli genome is 4.6 million base pairs, right? In that 4.6 million base pairs, suppose you want to say you want to identify the lag Z operon, which is around 4 kilo base pair. You can imagine it's typically like a needle in the haystack, right? You have lots of other sequences along with lag Z, but you're interested only in lag Z, right? So if you look at it, we need to design specific oligos for the lag Z region, okay? And then you melt this double-stranded DNA, okay? And there is a terminology called TM, or melting temperature. Melting temperature is a temperature at which at least 90% of your molecules will be in an open state. That means the double-stranded DNA bonds are broken. Okay, the hydrogen bonds are broken, but the phosphodiester bond is not broken. Right? The hydrogen bonds are broken. The hydrogen bonds between A and T, the hydrogen bond between G and C is broken, but the DNA retains its integrity. That means the double-stranded DNA becomes single-stranded DNA, but doesn't, become nucleotides or anything like that. Okay, and once you have, two strands, when they separate out, you actually have increase in the absorbance at 260 nanometer. This is called as hyperchromicity. Okay, we are not interested in this process, but I'm just telling you that when a double-stranded DNA becomes single-stranded DNA, the UV absorbance increases, and this process is called as hyperchromicity. Okay, and so after you denature, you are trying to anneal. Okay, annealing essentially is nothing but hybridization of of the corresponding sequences, complementary DNA sequences, which are also, which, as you said, it could be called, people call it as primers, some people call it as oligos, but these are sequences essentially which are complementary to the region that we are trying to amplify. Okay. So the primers anneal to the denatured double-stranded DNA, which has now become single-stranded. Okay, and it all depends on the some primers are extremely GC rich because of the sequence. So you need to have little higher melting temperature. And then for certain oligos which are extremely AT rich, you will be using a lower melting temperature or annealing temperature. Okay, and you will anneal these oligos to the genomic DNA, and then after you are done with that, you will start going for extension. Extension is nothing but the synthesis of a new piece of DNA by DNA polymerase, which essentially does this by adding the complementary deoxyribonucleotides in the 5 prime 3 prime direction. Okay, essentially use DNTPs. That means you will have DATP, DTTP, DGTP, DCTP, all the 4 nucleotides, okay, which are polymerized by the DNA polymerase, and you essentially have a new strand. Okay, so these are the typical cycling conditions. You start your denaturation at 95 degrees centigrade, and then you will do your annealing anywhere between 50 to 60 degrees centigrade. and then you will go for an extension at 72 to 75 degrees centigrade. Although it says 75, the temperature can vary anywhere between 72 to 75. Okay, depending on the polymerases that you might choose. Okay, and then this process continues for certain cycles. It could be 25. Typically in PCRs, people who do 25 cycles. Some people go for a little longer, 30 cycles, 35 cycles, but essentially by the 25th cycle, you will have enough product that you can use it for your routine purposes. Okay, so this is, the temperature from 90 or 95, it falls down to 50 degrees centigrade where annealing happens, and then you have an extension at 72 degrees centigrade, and then you have an increase to back to 90 or 95 degrees centigrade where the denaturation happens. Okay, the speed with which this temperature falls from 90 to 50, and then from 50 to 72, and then from 72 to it becomes 90, is called as ramp time. Okay, ramp time. So typically, in these PCR machines, it goes in a matter of few seconds. It goes from 95 to 50 degrees centigrade. Okay, it essentially cools down, and then from 50, in a matter of few seconds, it again increases to 75 degree 72 or 75 degrees centigrade, and from 75 it again goes back to 95. Okay, this process of heating and cooling is essentially is drawn from the laws of physics, which is called as Peltier effect. Peltier, Peltier effect, which essentially uses the same circuits for heating and cooling. Only that the direction of your current is changed, and when the direction of current changes, either you have heating or cooling, depending on the change of current. And this is what is called as Peltier effect. People didn't appreciate what that Peltier effect meant, but when it came to PCR machine, it became very, very apparent that Peltier effect is something that is so useful. You can use the same machine to, or same this thing, to heat and to cool down. Something cooling down from 95 degrees centigrade to 50 degrees centigrade using the same current is something unusual. Okay, no change in any component. Okay, so these are the basic principles. So this is what happens. This is, let us say, your genomic DNA. When you heat, when you denature, the two strands separate, and then you have a short primer which is complementary to this region, right? It goes and attaches very specifically only to this region. It will not attach anywhere else. And then you have a polymerase that can extend in the 5' to 3' direction. Remember your classes in basic molecular biology, whether it is transcription or translation or even replication, everything happens only in the 5' to 3' direction, right? So this is 5', 3', and then you have this new this thing template, new strand that has formed, new DNA strand. This again goes for, the second cycle goes for denaturation. You have primers getting synthesized, primers annealing to it, and then the DNA synthesis happens, and then eventually you have, a multiple number of copies. So remember, this is your original genomic DNA. You only have one copy, but then you have replicated, your PCR product many more times using this as a template. So the first time this serves as a template, but every subsequent cycles, every one of these products will serve as a template. That is why you see an exponential increase in the copy number, right? So this is simply given by a mathematical expression, which is 2 raised to the power n, and n is the number of cycles. Okay, when n is 25, then you have these many number of copies. Although you started with just a single copy, you just start with a single copy, but when you have 25 cycles, at the end of 25 cycles, you will have these many copies. And at the end of 30 cycles, you have literally, millions and millions of copies. So you are essentially starting with something. This, if you were to try to quantify or try to visualize, you may not be able to even see it. But then by the end of this, 25 cycle or 30 cycle, you can quantify them. You can see them on gel. Okay. So how do people typically, quantify the PCR thing? Basically by, okay, before that, how this entire process happens. It happens because you use enzymes like Taq polymerase, which you're very well aware of it, right? Taq polymerase. There are many different DNA polymerases are used. Essentially, if you look at the fundamental thing, they're all thermostable. That means they are extremely, all our enzymology tells that when you heat any enzyme, it loses its activity, right? But thermostable DNA polymerases from organisms that are in hydrothermal vents, where they survive extremely hot temperatures, those enzymes, those DNA polymerases are capable of working at extremely high temperatures, right? So but they need certain buffers, right? And very, very important, in all these PCRs, magnesium chloride plays an extremely crucial role. Okay, many a times, all these things come as optimized buffers. Okay, so the buffer generally has tris HCl, has KCl, has magnesium chloride, and some of them will have a small amount of gelatin to stabilize the protein. Okay, and you will have DNTPs because that is what is going to be used for extending the primers. Okay, and then you will have your template DNA and your primer. So these are typically the components of a PCR reaction. You will have the genomic DNA or cDNA, whatever you have, template DNA. You will have primers. You will have two primers. If you see this, you have two primers. So this is essentially called as a forward primer, and this is called as a reverse primer. So you will have two primers, okay, and you will have these DNTPs. You will have buffers and enzymes. Okay, typically the reaction volume is generally anywhere between 20 to 50 microliters. Okay, all these buffer comes as a 10x volume. Okay, 10x concentrated. So if you're setting up a 50 microliter reaction, then you will be pipetting about 5 microliter of your 10x buffer. If it is 20 microliter, then you'll be pipetting out 2 microliter of your buffer, 10x buffer. Okay, so that's just the ingredients of a PCR reaction, and this is how So this 25 cycle takes anywhere between 2 to 3 hours, depending on how fast your machine is able to ramp from 95 to 50 and from 50 to 75 and then again from 75 to 90. How fast your machine is able to heat and cool, depending on that it depends. Conventional PCRs take anywhere between 2.5 hours to 3 hours to complete it. Okay. At the end of it, how do you know whether your PCR reaction has worked or not? So this is done by running an agarose gel. Okay, which we kind of saw in the last semester when we did the practicals, right? So you will be running an agarose gel. Okay, and then so this is called as endpoint PCR. Endpoint. We don't know whether PCRs work, but the last step is to run it on agarose gel to see whether you got the PCR product or not. In this case, for example, PCR reaction 3 has not worked. Okay, there is no PCR product. This reaction did not work. Okay, on the other hand, PCR 1, 2, and 4, the PCR and 5, okay, they all work. Okay, this has multiple bands. Okay, it could be that when will you get multiple bands? For example, when you are using when you are trying to PCR out, say, some multi-gene families, right? They all will have very similar structures. Okay, and so the DNA probes may be binding to regions that are similar, and so you may get a multiple PCR product. Sometimes when your PCR conditions are not optimal, then also you will get multiple PCR products. So we need to optimize it. Okay, in this case, I'm not sure whether it is a suboptimal PCR or whether it is they have purposely done PCR to have three different PCR products. Generally, the end of PCR is only one single product, but you can also have multiple products. We'll come back and look at some of those things in subsequent classes. Okay. Now, along with your PCR products, you will also run molecular weight markers. So you will know, whether you are expecting what is the PCR product that you're expecting. Is it 1 kilo base pair? Is it 500 kilo base pairs? You will be able to know whether your PCR has worked or not. Okay. People also have, so these PCRs, for example, if you see, the biggest size is around 2 kilo base pairs, not beyond 2 kilo base pairs. Typical PCRs, when you do PCR reactions, the denaturation at 95 degrees centigrade happens for about 20 seconds. Okay, about 20 seconds, and then annealing happens for another 20 seconds to 30 seconds. Okay, and then when you do your extension at 72 degrees centigrade or 75 degrees centigrade, people do it for about a minute or two. The general rule of thumb is if you are trying to amplify a 1 kilo base pair fragment, then it is generally 1 minute. Okay. If you're trying to amplify a 2 kilo base pair fragment, then it is generally 2 minutes. If you're trying to amplify a 3 kilo base pair, 4 kilo base pair fragment, and all that, the general rule of thumb is need to have this extension time as 1 minute per kilo base pairs. 1 minute per kilo base pairs. Okay. So if you're trying to amplify a 5 kilo base pair fragment, then your extension will be for 5 minutes. Okay. But generally, Taq polymerase, is extremely good in amplifying up to 2 kilo base pairs, not beyond. Beyond that, it doesn't really do a good job. But people have had these different DNA polymerases, and now people are able to very easily and very routinely do PCRs up to more than 20 kilo base pairs. So, what an improvement from 2 kilo base pairs to 20 kilo base, 10 times more. Okay. So, these are specialized DNA polymerase mixes commercially available. So, somebody has put their heart and mind into making this, and then they are making a money out of it, right? So there is nothing wrong, as long as you are able to help people out, right? So people have done these long PCRs from different genomes, right? From lambda, from human, from E. coli, all these different organisms. They've been able to do this PCR and show that this PCR really works in all these different, or it doesn't matter whether it is human or E. coli or lambda, which is a bacteriophage. It seems to work very well. So, and this is called as fail-safe PCR premix. So these all come from a commercial organization which is going to charge you for that. You can do it yourself. There is no harm. You could do it yourself, but getting it from these people, they have gone through quality checks, and you may have to mix many different components. You could make some errors, unintended errors, and you may be losing time. So people would rather buy many of these things from commercial organizations and use this thing. So this is the first generation PCR machine, which itself was designed and done by Cetus Corporation. Okay, so they had a monopoly. Nobody could do PCR or use this PCR machine without their express this thing. So you need to have a agreement with them and all that. But eventually people made so many different PCR machine. Peltier Effect was something so common that people used it, and you now have many different kinds, all kinds of different kinds of PCR machines. So this is the first generation PCR machine where you can see on the monitor, where you could program on the monitor how long you want to hold your reaction mix at 95 degree. How long do you want to anneal? How long do you want to do extension? All this could be programmable. So this was your first generation PCR machine. This is something that is now routinely available in many different laboratories, including AIVA. We have this machine. This is your real-time PCR machine from a company called Biorad. Of course, there are many, many, many different companies that sell PCR machine. I've just put Biorad because I just saw that. Okay, so there is. It doesn't mean that only Biorad is selling. There are hundreds of companies that sell real-time PCR machines. So this is the screen. What was a small strip has now become a big screen. This actually continuously plots. Continuously plots how the PCR reaction is progressing. Okay, you now have advanced version of real-time PCR, which is called as a digital droplet PCR. Again, it happens to be from Biorad, but many other companies are also doing the same digital droplet PCRs. Okay, and these are very very useful in quantification, right? For example, in the current pandemic, for example, some people may be having a very high viral load, some people may be having a very minimal viral load. So we need to have some way by which we could quantify what each person has, what is the viral load each person has, and that is the uh, basis for real-time PCR. Okay, now three aspects of PCR that are extremely crucial to the success is its specificity, is its efficiency, its fidelity, and the fastness, the robustness with which these processes happen. The specificity is achieved because of the primers, right? In the whole genomic DNA, for example, of E. coli, you want to say only amplify the lag Z portion of it, then just having those primers, which is very very specific for lag Z, helps in making sure that it is extremely specific. It's extremely efficient because if you remember the our classes in basic molecular biology, we said DNA polymerase has this unique ability to polymerize several thousands of molecules in one go, right? In a matter of about 20 minutes, the entire E. coli genome is replicated, which kind of translates to about 4,000 nucleotides per second. So you can imagine the extreme efficiency with which these DNA polymerases are able to polymerize the nucleotides into DNA. Fidelity is something that is very very important. Fidelity means faithfulness. For example, when you try to copy a DNA, invariably, the DNA polymerases have an inherent ability to make mistakes. They make at least one mistake per 1 million copies they make. Okay, so invariably, your PCR may be a mix of, some molecules that may have some mutation in them. Okay, which is extremely crucial when you go for parenting, when you go for diagnostics for parental these things. We will come back and look at it, some of these cases during our applications of PCR. Okay. When people go for paternity testing, right, which is so common in India, when people when two different parents claim the ownership of baby, then paternity testing is done, and there it is extremely crucial to have extremely high fidelity enzymes. Okay. Okay, the best way to understand PCR is to consider all the reaction components and how they combine to produce the best results. So people used to spend a lot of time optimizing PCR, but nowadays PCR is so routine that people don't have to spend much time in optimizing, except suppose you have a new novel organism which you don't know, then you may have to do some initial optimization studies to make sure that you're able to do a successful PCR on those organisms. For example, we all know that Antarctica is something that is frozen probably for major part of the year, but people have even in those regions which are like permanently covered with snow, people have drilled deep, almost like several kilometers deep, and they find totally new life forms. I mean, like something that has never been seen in other parts of the world. Okay, and suppose you want to understand how these organisms are able to survive in these extreme cold conditions, you need to understand that means you need to understand their DNA, you need to clone, you need to sequence, and you need to understand how these organisms are able to survive in those extreme harsh conditions. So when you do something like that, we don't know what the genome is going to be. You may need to optimize your PCR reactions for these unknown organisms the first time. Okay. Each physical and chemical components of PCR can be modified to produce a potential increase in the yield, specificity, or sensitivity. This is what I said when, along with DNA polymerase, you may also add certain other components. For example, remember we saw that 0.1% gelatin is used. People also use something called as DMS or dimethyl sulfoxide, okay, which is an organic solvent. They use a small proportion of that, and DMSO seems to stabilize the Taq polymerase. Not only Taq polymerase, even other polymerases. So many people use DMSO. Okay, and people also can use other components, to increase the specificity. For example, running an agarose gel at the end of it, you may still be baffled whether how to quantify it. So people have gone back to see, instead of using regular DNTPs, can we use some modified DNTPs without affecting the incorporation of the modified nucleotide into the DNA strand. Okay, as we all know, DNA polymerases, or for that matter, any enzyme has the preference for substrates. And when you modify the substrate may no longer be used by the enzyme, right? So we need to modify the DNTPs in such a way that the DNA polymerase still accepts the modified substrate and polymerizes at the same rate. It is not that you modify the substrate and now the DNA polymerase is like working extremely slowly, 1/100th of the pace. That is not acceptable, right? So we need to modify it in such a way that the DNA polymerase still has a appreciable elongation or appreciable efficiency with which it can incorporate those DNTPs. Okay. Okay, so this is the Nobel Prize that was given to Carey Mullis in 1993 for in chemistry for doing this discovering this PCR. Okay, and as I said, I will send out the Scientific American article that was shared by that was written by him. It's an extremely, easy article to follow. There is nothing hard and fast in that. So I will share that. Please go through it. Okay. Any questions so far? Otherwise, we will start the real-time PCR module, and then we will continue with the real-time PCR module in our next class also.
Okay, sir.
No questions?
No, sir.
Okay. All right. So I assume that you all are aware of it. So, okay. My system seems to be okay. So is the screen visible now?
Yes, sir.
All right, thank you so much. So this is from Biorad, which is an instrument maker for real-time PCR. It also not only makes instrument, it also makes lots of these buffers and many other things which you could use for your PCR reaction. So we will just start with real-time PCR. Okay. And so what you see on this graph, what you see on this graph is many different, colors, right? Many different colors, and each curve, if you see, has a different starting point, right? Has a different starting. Just remember this, that you can essentially have different on these things starting points, and each of this has a significant meaning actually, and this is what is now used in terms for quantifying the this thing. Okay. Okay, so we will just see how this real time what is real time PCR, how does it work, what are the instruments that are used for real time PCR, how does a real time data look like, right? And how can this be used, real time PCR can be used in crime investigations, okay, in real time setting. This is just this is an example that Biorad has given, so I'm using it. But you could use real-time PCR for many different applications. You have many different applications. So in this case, they are talking about this. Okay. So what is real-time PCR and what is it used for? We all know that PCR process, as we discussed, is for the amplification of specific fragments of DNA. But real-time PCR is actually a specialized technique that allows PCR reaction to be visualized in real time. When we do the normal PCR, we don't know until the end of 25th cycle whether the PCR reaction has worked or not. As we saw, lane 3 did not have any PCR product, okay? And it is basically a failed PCR reaction. But in this case, with real-time PCR, you will be able to visualize in real time as the reaction progresses, okay? And so basically real-time PCR allows us to measure extremely small amounts of DNA in any given sample. Okay, so that's the biggest. So given the current pandemic, you can imagine that they probably take a nasal swab or a oral swab. Okay, and if you see the tiny cotton piece, a lot of it is going to stick to that cotton piece. But a teeny tiny amount of the virus or whatever material comes into the solution, and that is what is going to be used for amplifying the presence and detection for the presence of viral DNA or viral genome. Okay, so real time PCR has now has wide applications and has become like one of the essential tools in molecular biology, and it is used for gene expression analysis. For example, it is used very commonly in cancer research when people want to know what are all the different genes that are responsible for cancer. For example, liver cancer versus lung cancer, the expression profiles are very different. So they want to know what is it that is a driver for lung cancer, what is the driver for liver cancer. Many different these things can be done with real-time PCR. People use it for drug research. For example, if you give a drug and suppose the certain genes expression comes down, then what does that mean? Does that mean that it has a good therapeutic effect, or does it mean that the cells are responding? So some of those questions can be used. Okay, disease diagnosis and viral and management. We are so aware. We are so aware of quantitation of viruses in the current pandemic. Real-time PCR is the backbone of diagnostics. You can do food testing. For example, to look for the presence of genetically modified organisms, you could very easily do PCR. You will know. For example, I'm sure you are aware of the controversy over BT brinjal and BT cotton and so many other recombinant DNA products. So you could very easily simply do a PCR and find out, does the plant or any other product, does it have any other organism's DNA? Okay, animal and plant breeding, it's useful in animal and plant breeding. You could increase the copy number of certain beneficial genes so that it can have a benefit. Okay. These are just, it is useful in archaeological observations. For example, you found a new, archaeological site and let us say you found some bones. You could simply come back and, extract DNA, and then you could simply do a PCR to find out, what was the genetic makeup of that. Is it a human bone, or is it an animal bone, or is it something else, right? Even for example, the Siberian frost, where the many animals were frozen, people have used this PCR to clone the genes. Okay. Okay, this just gives you an example of how it is used in cancer, right? It says real-time PCR-based expression profiling of BRCA1 induced genes in primary breast tumors. As we all know, BRCA1 is one gene that is primarily responsible for breast cancer. So somebody is interested in understanding the mechanics of this BRCA1 gene. How does it drive breast tumors? All those kind of stuff. So we could essentially do this Another one is HIV treatment. Although it says HIV, you could simply replace HIV with COVID. You will still have essentially the meaning remaining the same. Okay. So here it says that. So this was in Journal of Clinical Microbiology. It says real-time reverse transcriptase initiated PCR assay with single copy sensitivity for HIV type 1 in plasma. It means even when you have extremely low copy number of the virus, you can detect it, and you could probably start treating the patient even before they start showing the symptoms. And essentially, I think that is typically what is being thought of even the current pandemic, that if people could be diagnosed at a very early stage, even before they show symptoms, then you could probably block the transmission. That is something that is we are slowly moving towards that. Okay, that means if you can detect through real-time PCR even extremely small amount of copy number, you are going to be better off, right? This is another one which talks about determining the percentage of genetically modified organisms in the GMO in food. Okay. And so that is the seed. This is your wild type DNA. This is the genetically modified organisms DNA. You could simply make the DNA preparation out of this and see how much of GMO it carries, genetically modified organisms DNA it carries. Okay. So many different things. Okay. Probably I would like to stop at this point and see if there are any questions. If there are questions, we can take it up. Um, Otherwise, we will continue with this on Friday class. How does real? Because I think it's better to start this in a new session. Any questions? So, what other areas where PCR can be used? Any idea? Where all we can use PCR? Vivek?
Sir, can it be, can I answer?
Yeah, please.
I'm not sure if this is right, but then I think in 12th we learned of a case where it can be used in, forensic sciences. So in a crime scene, let's say you
Mm,
find a
mm,
hair sample or a blood
mm,
sample,
mm,
then it
mm.
can be used in DNA fingerprinting to find out whose it is,
Extremely,
you
yes,
know.
extremely good example. So that is the power of, see, people used to use, just fingerprints, right? At the crime scene. From fingerprints, we have now moved into DNA fingerprinting. We are now moved into molecular fingerprinting. So that is enabled only by PCR, right? Very good example, Manaswini. Anybody else? Where else can we use? Where else can we use? Adish?
So can it be by detecting any mutation?
You could use it for detecting mutation. Very good, very good. These are all specialized applications, but nevertheless, you could use it for detecting mutations. Very good. Anybody else?
Yes. So like the process of PCR, we can literally multiply anything. So it can be used in almost anywhere if the conditions are there.
Yes. That's the purpose of PCR. You are going to clone, and you could essentially from starting with literally nothing, which you cannot really quantify. You are going to achieve, really a quantifiable this thing, product, PCR product. Okay, right. So. Keep thinking. Acha, I have mailed the gene cloning PowerPoint slides, till the last class, till the animal cloning thing. Okay. The only thing that I've not done is that FDA website, which I thought I will do it subsequently, which talks about therapeutic cloning and all that. Okay. Maybe I will mail that soon. But The otherwise the entire slide deck has come to you. Please let me know if you all have received it or not. This. All right.
Sir,
Yeah.
I have a question.
Yes, Shakti.
So, sir, like you said, in real-time PCR, we can get to know like our third reaction has failed. So,
Mm,
sir,
mm.
do we only get to know, or we can also by some means correct it, like in between,
No,
to make
once.
it worse?
But see, the thing is, when we do PCRs these days, we do multiple PCRs. Like for example, let's say COVID. Let's take the example of COVID pandemic. When you have taken the example of COVID pandemic, let's say these are all done in high throughput fashion. That means they may be detecting about 20 or 30 samples at a stretch, or in extreme cases, the reaction is done in 96 well plates. All of you have seen 96 well plate, right?
Yes, I
Ah,
saw
so,
in
so these, okay, anyway, when you come, we will show you a 96 well plate, but people have gone beyond 96 well plate. Now you can do even in higher numbers. Okay. So these are extremely small reactions. And, once a PCR reaction is initiated, if things are not progressing in certain cases, people don't go back to correct it. People would rather, for example, when you're testing for 100 patients, and if two or three patients are negative, you may not see any reaction. You may not see any PCR product in them, right? But that doesn't mean that the reaction is wrong. Okay. Unless when you go to an extreme case, when you have gone to, as I said, you have gone to, somebody has handed over to you some Antarctica sample and you're trying to do a PCR, but your PCR is not working, then you may be optimizing it for making your PCR work. But otherwise, a failed PCR reaction basically tells you that you need to optimize your PCR conditions. All right. Real time PCR, people don't go back to correct it. Okay.
Yes, sir. Thank you.
Any other question? Okay.
Sir, we can
Yes.
use PCR
Yes, you.
for to find out phylogenetic relationship between
Very good. Very
species
good.
too, right?
Very good. Extremely good example. People can use, PCR. For example, people could, let us say, many different organisms. You have a collection of bacteria, and let's say all of them are able to use, for example, say lactose, and you want to know how the, lactose utilization genes, do they look the same or do they look different? Say, for example, between Pseudomonas, Klebsiella, E. coli, Shigella, right, and Salmonella. You have a bunch of bacteria, and all of them are able to grow very well. And so you want to know how did they utilize, right? So you could simply, sequence and find out, how the genes are related to each other, how these organisms are related to each other. Very good example. Very good example. Okay, right. So we will. Yes,
Okay,
Yukta.
sir.
Anything? All right. So we will meet on again on Friday for this class. And as I said,